Standard targeted cancer therapies are designed for extreme precision, hitting a specific protein like a sniper rifle. But cancer is biologically adaptable. When a drug blocks one pathway, the tumor often mutates, finding an alternate route to keep the signal for growth moving. This cellular workaround is a primary driver of long-term multi-drug resistance, where a treatment that once worked suddenly fails. Overcoming this adaptability requires a network jammer, a compound that can strike multiple survival pathways at the same time to prevent the tumor from rerouting. Research has identified a naturally occurring flavonoid called Fisetin, found in foods like strawberries and apples, that functions as this type of broad-spectrum disruptor. In lab models, Fisetin doesn't just hit one target. It disrupts a massive network of oncogenic pathways, including Wnt β-catenin, mTOR, PI3K-AKT, and the inflammatory enzyme COX-2. By neutralizing these multiple defense routes simultaneously, Fisetin provides a layer of disruption that complements the focused precision of modern synthetic drugs. Inside the nucleus of every healthy cell, tumor suppressor genes act as the body's biological brakes. To grow unchecked, cancer cells actively switch these security genes off, particularly two known as p53 and PTEN. Studies show that Fisetin induces the expression of p53 and PTEN, turning these dormant systems back on. Once active, they force malignant cells, especially in colon and prostate cancers, into apoptosis, the cell's natural programmed death sequence. Zooming out to the cell membrane, we see the external signals the tumor needs to fuel its rapid growth. The PI3K-AKT signaling pathway is the primary engine cancer uses to coordinate rapid cell division and the recruitment of new blood vessels. Fisetin intervenes by downregulating the PI3K-AKT pathway, jamming the signal before it reaches the command center. This causes two critical failures. The tumor can no longer divide rapidly, and it loses the ability to trigger angiogenesis, stopping new blood vessels from supplying nutrients. By severing both the communication and the fuel lines, Fisetin works to isolate and starve the malignant growth. For cancer to metastasize, it must secrete enzymes called MMPs to chew through healthy tissue so the cells can move to new organs. Fisetin is a potent inhibitor of these MMP enzymes, essentially freezing the tumor in place and reducing its ability to invade surrounding tissue. In melanoma, Fisetin has been found to override MITF, a protein that regulates how aggressive and invasive the disease becomes. However, its most significant clinical potential may be in the area of drug resistance. Over time, many tumors build impenetrable defenses that pump chemotherapy drugs back out of the cell. Fisetin can alter the permeability of these resistant membranes, allowing chemotherapy drugs like doxorubicin to breach the cell and begin working again in pancreatic and sarcoma models. This makes Fisetin a valuable clinical partner, as it can help restore the effectiveness of conventional treatments that the cancer had previously learned to ignore. Beyond active treatment, Fisetin offers a protective role against environmental carcinogens, such as those found in cigarette smoke and industrial exhaust. In animal studies, adding Fisetin to the diet significantly reduced the development of cancerous lung lesions caused by these types of toxins. This dual capacity to neutralize environmental toxins while simultaneously disrupting established signaling pathways bridges the gap between prevention and therapy. As we move toward a model of integrative oncology, the goal is to combine the speed of modern medicine with the multi-targeted complexity of plant-based compounds to dismantle cancer from every side. Fisetin is a powerful plant compound, but your body destroys it almost immediately. To actually get it into your bloodstream, scientists rely on a trick called nanoencapsulation. Without it, you would have to eat 37 whole strawberries every single day to reach a clinical dose. Here is why. When a raw, naked Fisetin molecule enters your digestion, its low solubility and the liver's aggressive first-pass filtration completely shred the fragile molecule. But watch what happens when we wrap that Fisetin inside a microscopic fat-based liposome shell. This acts like a molecular Trojan horse, safely guarding it from harsh digestive enzymes. Because the lipid shell hides the Fisetin, it sneaks right past the liver's filtration system. And since the shell is made of the exact same material as your cell walls, it perfectly fuses with your intestinal lining, dropping the intact Fisetin into your bloodstream. So instead of forcing yourself to eat your way through 37 strawberries every single day, nanoencapsulation completely solves the absorption problem, condensing that massive dietary requirement into a single highly effective dose.